I'm both a clinician here and also work with a grant from the Wellcome Trust based at the Medawar Building on South Parks Road, leading a small research group. And 10 years to eliminate hepatitis B is basically a question that's set by the Sustainable Development Goals. So the United Nations has come up with, you may be familiar with them, a set of Sustainable Development Goals that apply to all sorts of questions around health and disease, climate, poverty, education, equity. So they're a really broad set of goals. But recently, and for the first time, viral hepatitis has been incorporated in these kind of international goals. And they've gone so far now as to say that we should be eliminating hepatitis B virus as a public health problem by 2030, which is quite an ambitious challenge because it basically gives us 10 years. And as I kind of show you some more of the data about this, it will hopefully become more apparent quite the scale of the ambition. So the first question is, can we do it? And the next question is, how can we do it? So I'm going to start off by just kind of introducing this virus. It's quite an interesting beast. It's a very tiny little virus, and we recently wrote this paper where we headlined it as being unique, tiny and misunderstood, which are basically three adjectives that remain quite true about it. So there are very few other viruses that have the same kind of biology and life cycle. It's one of the smallest pathogens, human pathogens, and there are definitely still some quite big holes in our understanding and in the data about this virus. So the backdrop to my project is that, as I said, I'm funded by the Wellcome Trust, and we've set up this new study recruiting patients, adults with chronic hepatitis B infection from these 3 sites. So we were lucky to already have a really good viral hepatitis cohort enrolling patients in Oxford, so that's the building we're in now, on the left. And then we've partnered up with teams in these 2 different centres in South Africa. So Bloemfontein is right in the middle of South Africa, and you can probably all put a pin in Cape Town right on the little point of South Africa. And the idea is that we recruit these patients into a cohort which will allow us to describe what hepatitis B infection looks like, look at how they do on treatment, look at the impact of HIV infection, which is obviously very common as well in South Africa. And then the nitty-gritty bit of the kind of science is to get the genetic sequence of the virus and of the patient and put those 2 together to see if we can understand some of the different outcomes of infection. And I'm going to explain a bit more about how that works and why we're doing it. But having kind of set up this kind of amazing scientific question that we wanted to study, we've kind of then taken a step back and thought, well, actually, there's quite a lot of other things in the mix. And if we just look at the immunology and genetics, we're missing a huge swathe of other questions that are actually really key and really integral to tackling this problem. And so there's actually this, I think a jigsaw is a good way of describing it, because all these bits fit together to try and inform ultimately what we do to make population health better. And so I'm going to use these jigsaw pieces to kind of set out the talk and describe to you just some of the little bits and pieces that we're doing to tackle the overall picture. So one of the central piece of the puzzle, if you like, which I'm going to start with, is understanding the virus. And I was kind of anticipating quite a diverse audience. I know there's some really specialist virologists here, but I'm anticipating that not everyone's in that category. So there's some kind of nice little definition. So I said I work in the Medawar Building, and this Nobel laureate is where that name came from, and he kind of came up with this nice description of how you might see a virus. And then there's also this kind of philosophical discussion about what a virus is, saying it's at the boundary between the living and non-living. So on the one hand, it's got a genetic code, it can replicate itself, it can have progeny, but on the other hand, it doesn't have any metabolism, it can't survive independently. So it's not quite a living being. So that's a really kind of interesting debate about what constitutes a living organism. And then this is a definition from the IT world, which is what's a computer virus, but of course it's derived directly from a pathogenic virus. And this also kind of says it very nicely, capable of copying itself and has a detrimental effect, including destroying data. So that's another way of kind of thinking about viral infection. So I mentioned to you that hepatitis B is tiny, and if you look at the kind of order of magnitude difference between hepatitis B and the human genome, it's kind of insane. It's many, many orders of magnitude difference. And we put together this chart which basically looks at most of the pathogenic viruses that we know that infect humans. You don't need to be able to read all of them, but I mean, they're sort of herpes viruses at the top, and then some of the things like common cold and flu in the middle. And hepatitis B is right down here on the scale, so it's almost it's almost the smallest pathogen that we know that can cause infection. So why is this a problem? Well, one of the kind of interesting things is that there are very diverse outcomes, and happily, coincidentally, they all seem to begin with C, which makes it easier to remember, for me anyway. So you can actually be exposed to the virus and then clear it. So your body gets rid of it, you make a good immune response, and it's gone. But it can also become chronic, by which I mean it becomes a passenger with you and you carry it with you, and it's not cleared or controlled. Control can mean that the virus is still present, but the immune system does quite a good job of keeping it under wraps. So we can detect the virus in our patients, but it's not particularly causing any obvious disease. It may be transmissible, or it may be at very low levels where it's quite unlikely to transmit. And then, of course, the things that we're worried about are cirrhosis, which is liver scarring, so inflammation of the liver leading to scarring, and liver cancer. And I'll show you some kind of quite alarming data about what's going on with liver cancer rates. And then the final C is cure. And cure's got a question mark by it, because at the moment we're not in a position to effect cure. So we can give treatment, but we can't actually cure the virus. And in a way, that's the holy grail of all of this stuff, is can we get to a point where we can give treatment that could actually eliminate the virus? So this is the kind of process that I've described, really, of someone with hepatitis B infection going from a healthy liver through to scarring and fibrosis and potentially to liver cancer. And it's also interesting that liver cancer can arise directly from what looks like healthy liver tissue. So this virus is directly oncogenic. It's directly causing cancer, even without that process of inflammation going on. So there are some really interesting pathways there that remain incompletely understood. And the other interesting thing about the biology is that the likelihood of getting into this chronic phase of infection depends on when you're exposed. So if you're exposed in early life as an infant or a baby, you're highly likely to end up with chronic infection. So you go on into potentially this pathway. If you're exposed in adulthood and you're an otherwise healthy adult, you actually have a very high chance of clearing the virus from your system. And children fall somewhere in between the two. And this is really interesting, and we don't properly understand what accounts for that difference. Why is it that children are so much more vulnerable? But what it does mean is that in many parts of the world where infections transmitted from mothers to their babies, you're getting these transmission events that then lead into a burden of liver disease in the population later in life. So I always think this applies very well to hepatitis B. An inefficient virus kills its host, a clever virus stays with it. And hepatitis B epitomizes that quite well. This picture in the background is actually hepatitis B in the blood, and it forms these kind of long filament kind of particles just made of the surface protein as well as the actual intact virus particles. So we know that this virus is a kind of passenger of humanity, and when I kind of reflect on how it can be that such a tiny virus it feels like we should be able to figure this out. Why is it that we can't? I think one of the things is it's so well adapted. It's been a passenger of human populations for as long as human populations have been around. And we also know that related viruses circulate in birds and in some small mammals and in some non-human primates. So these viruses are very well adapted and sort of integrated into human and animal populations. And it's interesting, I like this quote, the Bronze Age, fine jewellery, beautiful art, and hepatitis B. So in fact, some sequencing that's now going on of these kind of archived human remains is actually finding hepatitis B viruses. So these fragments of ancient viruses are now coming to light. So we know that this has been a passenger with humanity. And so it's really very recent that hepatitis B was actually kind of discovered and understood and characterised by Baruch Blumberg So he first of all described that little protein in the blood. It was called Australia antigen at the time because it was found in the blood of an Aboriginal Australian. And that discovery led on to the development of a screening test for infection, so looking for that protein in blood, the recognition of the relationship between this infection and liver cancer, and then the development of a hepatitis B vaccine. So that's kind of worthy of the Nobel Prize that he was subsequently awarded. So where are we up to now? You'll be happy to know I'm not going to explain this whole replication cycle, but we had to put this together for a journal article and we actually struggled to do it, which kind of tells you first that it's complicated and second that there are still some gaps in our understanding of how this all works. But I'm just going to point out two things about this which are interesting. The first is that, unlike lots of other viruses, hepatitis B makes its own little mini chromosome. This is the nucleus of the infected liver cell. So it sort of archives a copy of itself in the infected cell. And that's basically the crux of the reason why it's so hard to eliminate. So the immune system can't necessarily see that. We can't currently tackle it with drug treatments. And that's a reason why you get this persistence of potentially many decades of chronic infection. And then the second thing to point out here is that there's an enzyme in the life cycle which is called a reverse transcriptase, which is a close cousin of HIV. And we actually use drugs that are used in HIV as our first line treatment for hepatitis B. And the reason for that is this relatedness. And actually, hepatitis B often gets put together with other viruses that are called hepatitis, like A and C and D and E. But if you look at the biology of it, it's actually much more similar to HIV than it is to other hepatitis viruses. So again, sort of thinking about the biology and the nature of this kind of infection, it's interesting to just think about the way it manifests in the human host and how we measure that. So we can do lab tests that measure the number of viruses in blood, and I'm not going to ask for a show of hands, but you might just kind of want to pick where you think how many virions can be found in 1 ml of blood on that scale. And the answer is, in fact, over 100 million in 1 ml. So in a kind of 10 ml syringe, you've got 1 billion virions, which is kind of hard to even countenance, isn't it? But you can imagine that the liver is absolutely churning out virus in some individuals with infection. But as I've said, people handle the virus very differently. And in some instances, the virus persists at a really low level in blood. And in that kind of instance, we often see it without any evidence of liver damage or liver inflammation. So one of the things that we've done is we've looked at patients in Oxford, and we've just looked at the distribution of these viral loads. So if you take our whole population of patients with hepatitis B and you plot how many viruses each of them have in their blood, you get this distribution. So the average is around 3 logs, so that's about 1,000 copies per mil. But you can see this peak, and this is those really high ones that I just showed in the last slides. You get this interesting peak. So there are kind of 2 peaks, one quite a low level and one very high level. And although this has been sort of recognised for some time, we don't really understand what's going on there. So what is it about the virus or the host who's infected, or the interaction between the two of them, that puts people in these 2 different camps? And can we think about treatments that move people from this end of the scale down to this end, which we know will reduce the risk of transmission and it will reduce the risk of chronic liver disease? So one of the things to kind of reflect on about the biology is how we, first of all, understand the reasons for this distribution and then try and modify it with treatment. So there's a project that I've been involved with through the NIHR. So this is led by Ellie Barnes, who's a hepatology professor here, and a number of others also involved. So basically the aim of this project, it's a UK government investment putting money into this programme that basically says what we want to do is we want to take routine NHS clinical data. So you can imagine a hospital like this is churning out, vast amounts of clinical data every day from our various laboratory services. And we're going to use a kind of automated IT pipeline to collect that and clean it and anonymise it. And then we're going to package it up in a way that it can be reused for research. So that all of that data that we're generating anyway gets repurposed and anonymised and can then be used to feed into research questions. And that allows us to start doing these things like exploring the viral loads of our patients, and then we can look at other laboratory markers of infection and so on. And so this is has been done now in Oxford for viral hepatitis, and there's now a plan to roll out. In fact, it's kind of in action now that this is being rolled out to other centres across the UK, with the aim that we'll then be able to kind of collate much bigger data sets, which obviously give us much more power to look at these kind of questions. And viral hepatitis is one theme, but there are several other themes that tackle cancer and heart disease and so on. So hopefully this is something that we're going to get increasingly better at, although I must say the magnitude of the challenge is huge. It sounds really easy, doesn't it, when you kind of present it as a flow diagram, but there are lots of challenges to doing this well. But one of the things these data has allowed us to do is to start with what we observed in some of our patients in clinic, which is seeing the patients who do spontaneously clear that surface antigen, that protein, hepatitis B protein from the blood. And that's the closest that we currently get to saying someone's cured. So it's not a drug-mediated cure, and we can't necessarily eradicate that little mini chromosome from the liver, but we do see these instances where people clear all evidence of the virus from their bloodstream so that we can no longer detect it. And it only occurs in a minority of cases, so about 1% of people a year, 1% of adults with chronic infection a year fall into this category of clearing the virus. And as a result of collecting all these clinical data, we've now been able to map that to see what clearance looks like. So this is a timeline in days here. So it's showing you the kind of trajectory of clearance from the blood. Now, again, this is a clue about clearance of hepatitis B infection. It tells us that it's possible. There are immune responses, presumably, that are responsible for doing this, but we currently don't have insight into what those are. So this kind of data, collected from routine NHS clinics, allows us to start to address some of these questions about who are these people who clear, and what are the kind of underlying immune events that underpin that. So another kind of interesting avenue that we've been able to explore is actually with a local biotech company called Oxford Nanopore Technologies, and they've set up this new sequencing platform. So this is a way of generating sequences of viruses or other pathogens. So the way it works is it's a small device. I've got one in my bag, so if anyone wants to see, then you come and ask me at the end and I'll show you. But it's a small device. It's much like a mobile phone or a chunky USB stick. And you do a bit of sample prep, and then you load your sample into the device with a pipette. And then basically you plug the device into your laptop or computer, and then in real time you get sequence appearing of whatever's in your sample. So the way it works is that it will feed a strand of the DNA or RNA through this pore, hence the name nanopore, in a membrane. And as the strand of nucleic acid goes through that pore, it measures the electrical signal. Okay, and depending on what base, so these are A, T, C or G, that's the sequence of the DNA, depending on what's there, you get a change in the electrical current. And so at each position, you get an electrical readout. And so you can generate in real time the sequence of whatever's in there. And in our case, we've been trying to make this work well for hepatitis B, but we've also been using it in the hospital microbiology laboratory here to look for influenza. So looking at throat swabs that are coming in at this time of year now, we're starting to see influenza, and we've got this working quite well for diagnosing flu. And there are several advantages. One is you get a very long read, so you get a long strand of DNA or RNA. And the second is whatever's in there, it will read. So the person may not have flu, they might have rhinovirus or adenovirus or metapneumovirus or anything else. So this is, you don't have to set it up to look specifically for what you're looking for. You can diagnose whatever's in the sample. So it's a really kind of neat technology. It's portable, so you can put it in your rucksack. So my colleagues from the Medawar have gone off around South America with this in a minibus sequencing Zika virus. So it's an amazing kind of step change in what you can do out in the field, what you can do quickly. And of course, as well as diagnosing the bug, you can get all sorts of information about what's in that genetic code. So studying genetics was another bit of my jigsaw puzzle. So I've already embarked on talking about this. So we collect blood from our patients. we're interested in Hep B DNA, so we do some preparation steps to the sample to enrich that, and then we put it through nanopore or another sequencing platform, and we get our genetic sequence out. And this can give us lots of insights into the virus, so it can tell us about diversity of infection. So these are little families of viruses that are all slightly different, and that may have an outcome, it may influence the outcome of infection. It can teach us about the structure and the biology, so a bit more about that life cycle and understanding the structure of the virus. It may link into answering questions about liver disease, so those questions I asked about who does or doesn't get these different forms of liver disease. There might be clues in the viral sequence that tell us who's likely to transmit virus and what are the risk factors for transmitting and what kind of viruses might be transmitted. And it can also certainly give us information about responses to treatment. So if we give antiviral drugs, then we might be able to determine from that genetic sequence who's likely to respond or not. So the other side of studying genetics, I've talked about the virus, but the other side of it is host. And if you like, it's a bit of a seesaw. So there's going to be a balance. The outcome is going to be mediated not just by the genetic sequence of the virus, but also the host. And we haven't got as far as doing this bit of our project for hepatitis B yet, but there's this very nice example in hepatitis C virus done by Azim Ansari, who's a colleague at the Medawar. So essentially what this map represents is the whole of the human genome sequence down here and the whole of hepatitis C sequence up here. And then you do this for a whole population of people with hepatitis C. And then you look for relationships between changes in the viral sequence and particular sites in the human genome. And the ones in the colour, highlighted in colour here, are the ones that are statistically significant. So there's a statistically significant link between what's expressed by the virus at this position in the viral sequence and a polymorphism, a change in the human genetic code at this position. And they've gone on from this work to kind of interpret that, to find out new insights into the mechanism of immune control. So if you know what this gene is here, you can say, well, that's influencing the outcome of infection with hepatitis C. And so that's shed new light on the ways that hepatitis C is controlled or cleared by the host immune response. So as I say, this is a kind of currently still an aspiration for hepatitis B, but just gives you an idea, an example of why the study of genetics is important to unpick the outcomes of disease. Okay, so the next piece of the puzzle I'm going to talk about is burden of disease. So we now have these massive international collaborations called the Global Burden of Disease Study and big institutes that are interested in collecting data. And for the first time in 2016, we had this kind of headline saying that viral hepatitis is up there with AIDS, malaria and TB as a leading cause of death. And probably for most of you, certainly in the kind of medical literature, but also in the general kind of lay literature, I think these other topics get much more headline coverage. And viral hepatitis has really been very neglected. But as I've said, these are actually leading causes of disease. So this is years along the bottom, so year 2000 here, up to 2014 here. And this is a change in rates of liver cancer. So you can see this is quite an alarming trend, especially among men. We've got a really steep climb in liver cancers in men. And if you look at rates of most other cancers worldwide, they're actually going down. So liver cancer is really an outlier. And a lot of the reason for that is hepatitis B virus infection. So this is now a very common cancer worldwide and even more up there on the list of cancer deaths. And actually, one of the reasons for that is it tends to present late and we don't have good treatments. So there's actually a very poor prognosis. So if you think about the global burden of disease associated with hepatitis B, this is actually a really huge problem that hasn't been well represented in the headlines. Another way to look at the global burden of disease is this kind of map, which shows you the prevalence, so how common the viral infection is in different regions. And the red regions are those that are most affected. So you can see the whole of sub-Saharan Africa and lots of parts of Southeast Asia, that's over 8% of the population are, are actively infected with hepatitis B virus. You can also do a blood test to see who's been exposed to the virus, so that includes the people who've been exposed and then cleared infection. And about a third of us in total, a third of humanity has been exposed. So that's another reason why finding strategies to get to elimination by 2030 is a really tall order, just because of how common this virus is. I've focused on Africa because I actually started out working on HIV in Southern Africa, and I sidestepped into hepatitis B because I could see that this was a problem of equal magnitude in some settings, but not really having the same attention put on it. And if you look at Africa, there's all sorts of reasons why these populations are really vulnerable to hepatitis infection. And perhaps the most striking is that over 90% of cases are undiagnosed. So people just, the infection is silent and people just aren't able to access treatment. So the Sustainable Development Goals have actually now come up with these specific targets about how they want to tackle infection, getting infection down by these very specific numbers. So as I've said to you, this has been a kind of neglected topic to the extent that we wrote an article representing that. And if you look at the criteria for neglected tropical diseases, hepatitis B actually ticks all the boxes. So it's really a disease of poverty, which is why it primarily affects people in tropical and subtropical areas. It's associated with disease, stigma and discrimination. There are control and elimination strategies, and they're relatively neglected, so they haven't been well funded. And if you look at hepatitis B, we're really dealing with the tips of many icebergs. So undiagnosed infection is about over 90% of cases, which leaves us only 9% diagnosed, and then smaller proportions that are linked to care, actually attending and engaging with care, able to access treatment and have the virus suppressed by treatment. So you can imagine by the time you get down to here, it's not the tip of one iceberg, it's the tip of many icebergs. I was reflecting with some members of my group that the tips of icebergs doesn't seem a very appropriate analogy for a tropical disease and an African disease. And so we came up with this better analogy, which is the tip of the hippo, which is certainly more relevant for populations in sub-Saharan Africa. So it's the tips of many hippos, I suppose. So in terms of what's in the toolbox and what strategies we do, as I've mentioned, we have these drugs which are also used, some of them in HIV infection, but you have to be diagnosed in order to access treatment. We have a good vaccine, so that's Baruch Blumberg's vaccine that I mentioned at the beginning, but you do have to be able to, again, access it, and coverage has to be adequate. And then we can use those in combination and also with some other interventions to reduce vertical transmission, so that's preventing mother-to-child transmission. So since 1982, more than 1 billion doses of hepatitis B vaccine have been administered to children worldwide, so this is a huge initiative. And I'm interested to reflect on the idea that that now that we've got vaccines that can actually eliminate cancers. So this is a different example. This is that we can eliminate cervical cancer through vaccination against HPV, so that's human papillomavirus. But actually, if you just change a couple of words here, you could say we ought to be able to eliminate liver cancer by intensified vaccination against hepatitis B virus. And I think there needs to be much more advocacy around the idea of the hepatitis B vaccine as being an anti-cancer vaccination. So lots of countries have been rolling out vaccines. This is a nice poster from Uganda. Vaccines are like hugs, you don't want to miss one. And we've looked also at vaccination in Kimberley in South Africa. So Kimberley's the red dot right in the middle of the map there, which corresponds to this disused diamond mine, now flooded. There's not much else to see there, but there's a hole in the ground to go and look at. So when we started work there, we asked them about hepatitis B, and they said, well, you can look, but we don't have any hepatitis B here. And we then embarked on this study, and in fact we found that 11% of pregnant women there had active hepatitis B infection, and among those about 25% have this other protein in blood which suggests that they've got a high viral load and they're likely to transmit infection. So Kimberley was very representative really of lots of parts of Southern Africa, and saying we don't have any hepatitis B, but actually it's because it's not being looked for. They're not testing, and unless you test, then you don't find it until it presents very late with either liver cancer or end-stage liver disease. So these are some twins born in Kimberley, South Africa, and you have to think about all the kind of challenges of, premature twin babies in that setting, and what do we do to protect these children against hepatitis B, and are we doing it effectively? So having looked at the antenatal women, we then had a look at the children, and in fact far fewer children are hepatitis B infected, less than 1%. And then we looked at how many children are making good responses to the vaccine, and in the study group that we looked at, about 3/4 have evidence that they're protected by the vaccine when you do a blood test and you look at their antibodies. But the rate of that goes down among those who are HIV positive. So in HIV positive children, only about half, only about 50% have got evidence that they're protected by vaccine. And in all of them, the vaccine responses tail off over time, such that by the time they're about 5, they might be losing protection from the vaccine. So is this going to help us reach these elimination targets? Well, the answer is vaccine works really well, and it works really well in children. We've reduced thousands of infections worldwide by rolling out the vaccine. But it's not a complete solution. So we're still seeing cases of children infected, and there's still some worry about the tail off in antibody over time. So that's some thoughts about vaccine. And then, of course, I've said that we've got these drug therapies, but they don't cure the virus, they just suppress it. And to take treatment, we use these relatively sort of detailed algorithms or assessments for our patients. So we measure various blood tests, and we try and do some imaging of the liver, and some patients have a biopsy taken. And so this requires a laboratory and a clinical service to support it, and it's not really sustainable for lots of parts of the world. And it's interesting that only about 20% of patients currently meet these criteria for treatment. So 80% of everyone with hepatitis B infection currently doesn't meet criteria for treatment. But at the same time, I've shown you we've got these increasing rates of liver cancer and a burden of liver disease. So one of the questions that we've been thinking about is whether we should actually be treating more people, more of the hepatitis B infected population than we currently are. I'm conscious of time actually, so I'm going to skip relatively quickly through these slides, but this is showing some clinical data from our study group in Stellenbosch in Cape Town, and looking at the role of hepatitis B and HIV, so what's the impact of co-infection. And essentially what you see if you compare individuals who just have hepatitis B, which is on the left bar, compared to those who have hepatitis B and HIV, the outcomes are always worse in the ones who just have hepatitis B. And I think that's contrary to what we might always expect, right? You'd expect that if you've got both viruses, hepatitis B and HIV, that you would do worse. But in fact, the ones who have HIV tend to be the ones who are on treatment. Okay, and their HIV treatment also treats the hepatitis B. And so if you look at any parameters, these are all different ways of measuring liver disease. In all of them, the ones with just hepatitis B fare worse, and the HIV infected group do better. So really this is a piece of data saying if you're in care for HIV and you're on HIV treatment and you're being scrutinised and treated, you do better. So this is another kind of piece of the puzzle saying that maybe providing access to drugs for more people is going to be really important. So you can really regard this question about drug therapy as being a scales. There are pros and cons of thinking about extending treatment to more people, and some of these are quite powerful arguments, I think. So at the top of the pile on the plus side is saying we'll reduce liver disease and we'll reduce transmission. And those two things have to be high on our agenda if we're going to get to those 2030 elimination targets. And of course, there's caveats to doing that, and I'll talk a bit about some of these things, but the potential for drug resistance, the cost, the committing lots of people to lifelong therapy, those are all quite big things to do. But what I think we need to do is regard that scales as still unbalanced, and we've got to kind of work out some more of the evidence in each of these boxes before we can really reach a decision about whether we should be extending treatment. So this figure looks a bit alarming, but it's a map basically of one of the structural proteins in hepatitis B based on what we know about HIV. And it's just to illustrate the way that drug resistance can arise. So if the hepatitis B protein is configured in a particular way, but the virus then makes some substitutions in its genetic code, which are marked here, then it may be able to evade the effect of the drug. So the drug can no longer access the enzyme, or it can no longer bind, and you then end up with a virus that can escape from the effects of the drug. So it basically renders the drug ineffective. And in fact, it's now worrying, but we know that this can happen for vaccine as well. So there are now vaccine escape mutants in the same way. So Jolynn, who's my PhD student, who's actually in the audience, which is great, has done some work looking at hepatitis B drug resistance across Africa. And of course, you can't read all the individual results, but you can look at the spots on the map which show the sites where studies have been done. And I think the striking thing is all the countries where studies haven't been done, and the fact that each of these spots has really a very low number of studies. So 5 from South Africa, but lots of these countries only have 1 or 2 studies. So despite the fact that 8 or more % of everyone in sub-Saharan Africa has hepatitis B virus infection and liver cancer and liver disease rates are rising, we basically know very little about whether the virus is likely to be responsive to drug therapy or not. So we've done some work to try and map infection. Jolynn's nice map of Africa is one starting point, and we've also done a review looking at all the data we can get from different African countries, looking at how many are actively infected, how many have been exposed and cleared, and then the rest of the population are those that are susceptible. So we've done that in as systematic a way as possible, and you can then look at the relationship between being exposed to the virus at a population level and having active infection. So these are the data for the whole of Africa shown on one plot. And as you'd expect, broadly speaking, the more the population is exposed, the higher the rates of active infection. But the relationships aren't completely consistent, and you can also pick this apart by geography. So this is very broadly by region. You can see that there are broad differences across Africa in the prevalence of active infection, those that are exposed and those that are susceptible. And the reason for doing this is that it tells us a bit about who's being exposed and when. So that different relationship can point to a lot of infections in children or more infections occurring in adults. And it can also direct us with what to do with vaccines or what to do with drugs. So if you've got a very susceptible population, you might want to go in and vaccinate more of them. And if you've got a very exposed population, not many will actually be susceptible. And so you're better off not doing more vaccination, but going in with testing and treatment. So with collaborators in Oxford, we've developed this model. I'm not going to talk you through all the parameters, but it's basically a mathematical way of looking at transmission and the way that you might intervene in that. So you consider your population in these different groups in the circles, which are susceptible, vaccinated, acutely infected, chronically infected and recovered. And you look at the flow between those pools, and you look at how you might interrupt that flow using different strategies. And you can then apply that to say, if I do X intervention, how will it pan out in a population over time? So for instance, if we do infant vaccination and we do it in 100% in the turquoise here, then how will that impact on prevalence? And you can see it goes down very steeply and it eventually goes to extinction. But the key thing about this is to look at the scale on the bottom here. This is years after intervention start, 500 years. So are we going to achieve elimination by 2030 based on hepatitis B vaccine? Based on these data, no, we are not. And the reason for that is that there's such a big pool of infection in the population and that vaccine-mediated immunity is incomplete, that even with very high coverage of vaccine, we're not going to reach elimination. And actually, just to sort of verify that, there's various other studies that have looked at this. This is a group in London who've done a similar model, and if you look at their prediction for where we're going to be by 2030, far from going down, if we stick with the status quo, we're actually going up in terms of Hep B deaths by the year 2030. That is our current trajectory if we stick with the status quo. So you've seen the top half of this slide already. Vaccination is working in children. Please do digest that because it's really important. But also true, if we stick to the status quo, we are not going to reach elimination by 2030. And we had to work really hard to publish these two messages together because the journals thought there's a bit of a conflict here. But isn't it very important that the two messages get said together? You don't want to just say the vaccine's fine and that's going to get us to the elimination targets. You also don't just want to say vaccination's a disaster and it's never going to be successful. That both of these things, I believe, are true. So what is the best intervention? We've used the modelling strategy to look at other options, and the purple bar on this chart is the one to focus on, which says diagnosis and treatment. If we diagnose and treat infection, look, we can actually achieve the greatest impact from all of these potential different interventions, including vaccination. And so that raises the point that as well as being valuable as an individual intervention to prevent liver disease, that treatment can be valuable to prevent transmission. So briefly, just thinking about tackling poverty and stigma, I found this photo by chance on Twitter. It's the winner of an environmental photographer of the year this year. And it kind of takes some digesting, doesn't it, that is still the case in our modern world, that a child is drinking that kind of water, and that kind of says something about the magnitude of the challenge of tackling healthcare in some of these settings. But we've collected some impressions of what people are dealing with these infections in Kenya. So again, supported by Jolyn and others in the team. And the sad reality is that there's a lot of poverty and a lot of stigma. Patients come to hospital late because they don't have enough money, and most of them die. Healthcare workers scolded my husband for not taking his medicines, but they've tried to sell their home and their animals. So people are in desperate situations. And we've looked at this in Uganda. We found there's no word for hepatitis B in many of the local languages. There's lots of gaps in understanding. Many populations still use traditional beliefs and practice, some of which may be relevant and helpful, and others not so much. There have been some radio campaigns which provide good education, but generally people come to treatment too late, and that leads to these poor outcomes. So that brings me to the point of engaging and educating, and we've done some work to publicise this. So this was an article in Nature earlier this year where some of our own patients' voices and stories were represented. And then we've also tried to make some fact sheets to be used and translated into local languages to make this more accessible for our patients. So if you look at trying to kind of pull this together and answer the question about elimination by 2030, there's lots of conflicting voices, and these are just two examples. First of all, saying that with a concerted effort, most of the elimination goals could be met by 2030. But then another one saying every 30 seconds someone dies because of their hepatitis B virus infection. Isn't it time to reevaluate our strategy? And the image that's been used for that is these troops going over the top of the trenches. So in the kind of discussion and dialogue that's being disseminated around this, there really isn't a consensus about whether or not we can achieve these 2030 goals. And the reality of the truth is, of course, somewhere in the middle. And I hope that I've been able to present to you some of the reasons to be optimistic, that we do have this brilliant vaccine that really works, particularly in protecting children, that we've got safe and good antiviral drugs, that we can prevent transmission, and that we've got this advocacy through international goals. But of course, on the other side, there are all these challenges and gaps. We still don't completely understand the biology of infection. It's been very neglected, and stigma and poverty are major challenges for many of the populations where this is the most relevant. So I'll end by thanking some of the many people who've been involved in this. This is my research group in Oxford. You'll be pleased to see we have a dog and a baby represented as part of the team here in Oxford. Many individuals in our sites in South Africa, but particularly the two African teams represented here, and then lots of institutions and funders in the UK and further afield. So I'm sorry I've talked for a bit too long. I'm really happy to take questions, and I'm so grateful to you all for coming and being interested and enthusiastic about this. Thank you.